Polysaccharides. Polysaccharides are polymer of 10, 20 or more monosaccharide units. The polysaccharides can be classified into two types based on the presence of monosaccharides. They are homopolymers or homopolysaccharides. Second one, heteropolymers or heteropolysaccharides. Homopolymer composed of a single type of monosaccharides. Example, glucose or homopolymer of glucose. Glucons are homopolymer of glucose, means in glucons only glucose molecules are present in more number. Monons are homopolymer of monos. Monons are made with only monos with more number. Levons are homopolymer of fructose. More number of fructose molecules are present in the polysaccharides called levons. They are belongs to homopolymer or homopolysaccharides. Heteropolymers or heteropolysaccharides made of variety of different monosaccharides. They are also made up from a variety of different monosaccharides like glucose, monos, fructose, galactose. Like these different monosaccharides are formed in polysaccharides or polymer. They are also called heteropolymers or heteropolysaccharides. Heteropolymers or heteropolysaccharides. Polysaccharides are non-sugars giving more than 10 molecules of monosaccharides on hydrolysis. They may also polymeric anhydrides of simple sugars. They do not have sweet taste. They are amorphous substances. Amorphous substances means they are having different shapes. They're not proper structure, not proper shape, amorphous substances. And they are insoluble in water. They have high molecular weight. They form colloidal solution when heated with water. They do not exhibit any of the properties of aldehydes or keto groups. So polysaccharides structure, functions. Polysaccharides are complex carbohydrates made up of or made up linked monosaccharide units. Made up linked monosaccharide units. When a polysaccharide is made up of one type of monosaccharides of units, it is called homopolysaccharides or homopolymers. Starch and glycogen are polysaccharides used for energy storage. Other polysaccharides such as cellulose and chitin may be structural in functions. Now, polysaccharides or glycons. Polysaccharides are also called glycons. So here it is homoglycons or homopolysaccharides like starch, cellulose, glycogen, dextrins or inulin. These all are belongs to homoglycans or homopolymers or homopolysaccharides. Second one, heteroglycon. Heteroglycans are heteropolysaccharides or heteropolymers. Example, mucopolysaccharides. See, polymers have molecular weight from 2 lakhs and white amorphous products, glassy. Amorphous means they didn't have any structure. They are in white color, not sweet in taste, not reducing, to not give the typical aldose or ketose reactions, form colloidal solution or suspension. And the polysaccharides are having a great majority of carbohydrates of nature occur as polysaccharides. Chemically, the polysaccharides may be distinguished into homopolysaccharides, heteropolysaccharides. We know already homo means which yield on hydrolysis a single monosaccharides. Heterosaccharides means which produce a mixture of monosaccharides on the hydrolysis based on their functions, aspects, functional aspects. The polysaccharides may be grouped under two heads. One is nutrient or digestible polysaccharides. These acts as metabolic reserves of monosaccharides in plants and animals. Example, starch, glycogen, inulin means they gives energy, metabolic energy to the living organisms. Second one is structural or indigestible polysaccharides. They serve as rigid mechanical structure in plants and animals like cellulose, pectin, chitin, and also hyaluronic acid, chondroitin. These are the structural or indigestible polysaccharides. They are mainly cellulose, pectin, chitin. They are mainly helps in structure of the cell walls in the plants. Structure of cell walls in the plants. Now next, in that polysaccharides, in that homopolysaccharides, types of polysaccharides, one is starch. Starch is the major digestible polysaccharide in our diet. The storage form of carbohydrates in the plants. Source of starch: wheat, rice, corn, rye, barley, potatoes, tubers, yams, and another products. The two types of plant starch is present. They are called amylose and amylopectin. Amylose, amylopectin. See, it is a means starch is a homopolysaccharide. It yields glucose on complete hydrolysis. So it is a glucan. It is the reserved food material in plants. They are found in the cereals, legumes, potatoes, vegetables, sago palms, arrowroot of monihot or monita, and tapioca of monihot. It is a white amorphous, tasteless, soft substance. It is a amorphous. Amorphous means non-improper structure and tasteless, soft substance. It is means starch is insoluble in cold water, alcohol, and other, but it insoluble in the ether form, colloidal solution on boiling water. Mainly, the starch is insoluble in cold water, in alcohol and ether, but forms a colloidal solution on boiling with water. It gives, means starch gives dextrins during the incomplete hydrolysis. Dextrin used as a adhesive and for feeding infant because it easily digestible. Starch is made up of two structural different homopolysaccharide units. They are amylose and pectin, amylopectin. Amylose is 20% and amylopectin is 80% of the starch. Now first we discussed about here, it is amylose. Amylose, it is a homopolysaccharide with a molecular weight range from 10,000 to 50,000. It has 1,4 glycosidic linkages. See here it is 1,4 alpha 1,4 glycosidic linkages. The glycosidic hydroxyl group of one glucose unit is joined to fourth carbon of the next unit of glucose. It is water soluble and has a long unbranched chain unit. It gives maltose during the enzymatic hydrolysis. The enzyme is used amylase. It is considered to be an anhydride of alpha D glucose units. These are alpha D glucose units. It gives means the amylose gives intense blue color with iodine test. Intensive blue color with iodine. Now comes to the amylopectin. It is also homopolysaccharide with a high molecular weight of 50,000 to 1 crore. And it is a branched chain polysaccharide. See here it is branched chain polysaccharide. It is insoluble in water. It gives a isomaltose during the incomplete hydrolysis. In addition to 1,4 glycosidic bond, alpha 1,4 glycosidic linkages, and it has alpha 1,6 glycosidic linkages in the chain attachment. The average chain length, it is average chain length. Amylopectin is about 24 glucose units. Harvard proposed laminated structure of amylopectin. Mayer proposed the randomly highly branched structure. It gives means amylopectin gives purple color with iodine. Purple color with iodine. So these are the major components of starch. Now next one is glycogen. Glycogen, the storage form of glucose in the body, stored in the liver and muscles, found in tiny amounts in the meat sources, not found in the plants, not a significant food source of carbohydrate. So glycogen is a glycogen is a homopolysaccharide. Since it gives glucose on complete hydrolysis, so it is a glucan. It is the major reserve carbohydrate in the animals, so it is also called animal starch. It is stored mainly in the liver and muscles of all animals. Among plants, it is found in fungi and yeast, which are absence of chlorophyll. Absence of chlorophyll is also called devoid of chlorophyll. It is a white powder. It readily dissolved in water. The solution gives means the glycogen gives reddish brown color with iodine. Reddish brown color with iodine. It is a branched polymer of glucose with alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds. Alpha 1,4 glycosidic bonds, alpha 1,6 glycosidic bonds. See, the minimum molecular weight of glycogen found to be 50 crores of molecular weight. The actual weight of glycogen inside the tissue may be higher than of this. It is non-reducing sugar. It gives red color with iodine. Red color with iodine. The red color disappeared on the boiling and reappeared on the cooling. The liver glycogen supplies glucose to all tissues through the blood. The blood always contains 1% of glucose. When it exceeds 1%, the glucose transport into liver and it convert into glycogen. This process is called glycogenesis. When the blood sugar levels below 1%, the liver glycogen changed into blood glucose. This process is called glycogenolysis. Glycogenolysis. The muscle glycogen is utilized as the energy source during the muscle contraction and relaxation. The glycogen resemble amylopectin of starch chemically, but differ from the in the molecular weight of degrees of branches. It is the main structure of glycogen and functions of glycogen. And next one is inulin. Inulin is also called fructosan. It is also called storage polysaccharide. It linked with beta 1,2 linked fructofuranosis. Linear or no branched, lower molecular weight than starch, color yellow with iodine. Sometimes they didn't use any color. Sometimes they didn't use any color on with iodine. On hydrolysis, yield fructose. Source includes onion, garlic, dandelion, and Jerusalem. The inulin is a major source. Inulin is a major source for the storage food. Inulin is the major source for the storage food. It reserved carbohydrates in many plants. It is a homopolysaccharide. It is a homopolysaccharides. It present in many plants. They are also called non-sugars with 30 to 35 fructose unique molecules of units per molecule with molecular weight about 5,000. The 6 carbon atoms of cited are situated in the 5-membered furanose ring. It is formed by eliminating a water molecule from the glycosidic group of 1 carbon atom on carbon atom 2 of 1 beta fructose unit of the alcoholic group of first carbon of the next fructofuranose. So this linkage is called alpha 1,2 fructose. Sorry, beta 1,2 fructose. Beta 1,2 fructose. Beta 1,2 fructofuranoses glycosidic linkages. Beta fructofuranose 1,2 glycosidic linkages. On the hydrolysis, inulin gives a small amount of glucose in addition to fructose molecules. And it is a powder. It is a white powder, insoluble in cold water, but soluble in water, warm water to form colloidal solution. It does not give any color with iodine. Say already I'm saying only few times it gives yellow color with iodine, but does not give any color with iodine with the rest of the time. It is used for physiological investigation for the determining the rate of glomerulus filtration. So it is the main properties of insulin belongs to polysaccharides.